Okay. Alrighty. So at the top, what we want to write is where it got cut off is the cell cycle begins here. Okay. I hate that got cut off, but so the cell cycle begins here, and on the other side is mitosis. Now, this is some of the most important stuff that we do conceptually to understand, so we actually will take a break. We'll do the left-hand side and then the inside, then we'll take like a couple-minute break, and just so you can stand up, get water, kind of regroup, and then we'll do the right-hand side. And that seemed to work really well with everybody yesterday. So, all right, so the cell cycle begins. What I want you to picture is the cell. So we drew a skin cell, but it could be any body cell. And so if this is a human body cell, one of our skin cells or stomach cell, intestinal cell, hair follicle cell, whatever, what cell enters this? What's the chromosome number that we wrote? 2N. So it starts in like any old cell that you've got on your body. This is going back to Caleb's knee. So as his knee heals, we've got these cells that are around there that are having to divide to fill in the gaps. It's like a bunch of them just got torn off, and now they have to, all the surrounding ones have to divide to heal this and fill the space. So we're starting off, so for us, that's 46 chromosomes. If this was a corn plant, it would be 20. If it is a goldfish, I think it's like 92 or something crazy. It's a lot. So whatever it is, it has to be 2N. So here's a body cell. Okay. All right. So after G1, what happened to the cell? What happened during G1 to the cell? All right. So let's open the flap up, and we're going to put growth. All right. What doubled? Not the DNA, but something else. All right. So organelles doubled. Okay, so if we had 10 mitochondria, we have 20. We said you have double the organelles. Okay, so you need more space. And then what else were made? Yeah, proteins, protein synthesis. So RNA has read the different genes and is making the proteins that are necessary. So protein synthesis. So those are the events of G1. Okay, so then we go on to S. So this cell has gotten bigger, so it grew. So let's draw a bigger cell. Did anything happen to the nucleus, to the DNA in G1? No, right? But in S, what happens to the DNA? It doubles. Okay, so instead of 2N, what do we have, y'all? 4. So let's put 4N, 92. Chromatids / chromosomes. All right, so we have a bigger cell. The cell's grown in size. It has extra DNA. So inside that flap, that's what we put. We just put DNA replication. And I'm going to put in nucleus. I would put that if you don't remember where that happens. Just remember the DNA is inside the nucleus. Okay. All right. What happened in G2? Whoa. Now it's like monster cell is so big that this cell, if it doesn't go on to M and divide, it's going to pop. So it's got extra everything, enough to be able to create 2 new cells from this big giant cell. All right. Still 4N. Nothing's happened to the DNA. This just now has a lot of extra cytoplasm, and it's ready now to go. So on the inside of that one, we'll put more growth. Okay. Preparing for division. Okay. Or you could put preparing to split. Whatever helps you remember that. Okay. So in M, we call that cell division. So what do we end up with after M? What did we draw? Yeah, so it's divided in half totally after cytokinesis. So that's what we're going to draw. Okay, so we've got 2 cells. After that one has split. Now, if we just went from this cell that's 4N and it split, the nucleus split in half, how many chromosomes are we going to write in each of these, y'all? Right, so we divide it in half, 46, 46. What do you get if you divide 4N?
2N.
2N and 2N, right? So 2N, 2N. So we ended up with 2 cells. How do they compare to the one you drew in G1? And then I might draw mine a little smaller, but how does these cells compare to the first? That's it. So in this flap, we're just going to put cell division. Okay. Mitosis, cytokinesis. Okay. So that's where that's where the information stops for the quiz on Monday. So that's just a nice review. Okay, so there's a question you're going to see, I feel pretty certain, that shows a diagram that looks like this. Okay, so everybody got that so far? Yeah? Okay. The diagram, we'll do it on the back, and I'm just going to draw it. 2N, 2N. You're going to be asked, this is mitosis. And they're going to ask you, what is the result of mitosis? And you have to find the diagram. Why is this the correct answer? You get two cells the same as the first. What happened during S? If you flip back over.
DNA replicated.
Right. You don't know how many people say, that's not right, that's not right. 2N can't be split into 2N's. The math doesn't match up. But what you have to remember is DNA doubles. So that's where you go in and you're like 4N. That's right, DNA doubled, then it's split into 2 of the same thing. So that's why this is the answer. Okay? Now, this is important because when we look at sex cell reproduction, which you guys will be working on Tuesday, sex cell reproduction, after this, they split again. So we get 4. But they don't, they actually, if we split these, we'll get 1Ns, right? So that's how mitosis works. Okay, so if we flip to the center with mitosis, mitosis is making cells. So it's cell division that creates 2N or diploid. If you don't remember that 2N means diploid, I'd write the word. If you do remember 2N's diploid, I wouldn't worry about it. Okay, so the cell division that creates 2N body cells. All right, that's also called somatic cells. So they may use that word instead. So you see soma means body. That's why there are some mattresses called soma. And they're talking about, I guess they want your body to sleep well or whatever. So, but soma is body. So those are any of your body cells. We're going to skip who does it. Why? Why do you have to make more body cells?
If we don't, then it's probably going to go to the bone.
So what happens?
If you like body cells, if you don't, like skin cells, if you don't make enough, if you quit making it, then eventually it's not going to go away and you want to see your bones and muscles.
Oh, yeah. So you have to replace the ones you lost. You've got to heal. Okay, so let's put those. So why mitosis? Replace lost ones / heal. All right, that's so true. I mean, you got to do that. Okay. And you started off as a one cell. So why else do you have to make more cells? grow. And what else goes with that? What's development? Yep, development. Okay, and there's something that we can't do that some organisms do.
Like viruses or something.
Which reproduction is basically an extension of your body? Asexual. So yeah, mitosis would be asexual reproduction. Okay, so we'll talk about that on Monday. We'll get into sexual versus asexual, and we'll look at just some different types of asexual. And then on Tuesday is your sexual day. Okay, sounds kind of weird, but Sex Tuesday, I guess. Okay, so who has to be able to replace the ones they lose, or grow, or develop, or reproduce asexually? Basically, who are we talking about? What kind of organisms? Huh? Everything. Yeah, in some way. So all organisms will use mitosis for some reason. And it's kind of cheating a little bit because bacteria, technically, we just call it binary fission, but it's basically mitosis. And because that's how they reproduce asexually. All right, so when do they do that? That's just as needed, as the organism requires it. So we're going to put as it's required. Let's put to meet needs of organism for whatever reason they're using mitosis. Because we know you don't grow forever. You're not like this tree out here. This tree's still growing. They'll grow until it cuts it down or it gets diseased and it dies. But we can't grow. So, Absolutely. Yep. They'll get diseased and then you'll have to cut off those branches and you'll see different types of rot. The leaves will get fungal disease. There's a little black spots you'll see on leaves. So, yeah. A lot of fungus. There was one type of fungus called a blight, B-L-I-G-H-T, just like it sounds. It's called chestnut blight. It almost wiped out every chestnut tree in America. Wiped them out. So if you ever hear, and I don't see it often, but chestnut is one of the most expensive woods you can find in America. So if you ever find any kind of especially at old auction sales and things, people might have some old pieces of furniture that's chestnut. They're worth a lot of money. It's just because it wiped almost every chestnut tree out. So it's happened. Okay, Hal is going to be over here. So here's our break. Okay, so you stand up, you can get some water, you can kind of stretch, you can kind of move around. But just because we have to be full on for the next part. Okay, so stand up, take advantage of it. Do some jumping jacks, push-ups, plank. That was the yesterday, yeah. And it's still going to, I'm just going to let it run because it's harder to cut it off and cut it back off. And then we'll do the other side. That's where the colored pencils come in. Actually, in this break, why don't y'all go ahead and if you're going to do Ed Puzzle on your phone, then you can just use your phone at that time. Otherwise, if you're going to use a computer, you may want to get a computer. Okay, that would be a good time to go ahead and knock it out. If you have earbuds and stuff. Huh? Yes. Do you have a hall pass? Mm-hmm. Yeah. And you can actually go ahead and get into Ed Puzzle just to have it ready. I hope we get finished with the flip book. If not, if we get at least the first part, then we don't get the psychokinesis. That's okay.
I'm gonna finish the Ed Puzzle. I'm doing lunch.
Okay, that's fine. Yes, if you can get into your account. If not, I can't stop the broadcast yet, but I can look it up for you. Or you can rejoin if you don't remember your password.
Is the vacuum truck you listen to this?
It should be all the way at the bottom. Pushing to. Yeah, join it. Okay, well, let's go ahead and start this, and then I'll stop, so that way you have some time. But we may only get, we'll see. We may only get just a little bit done. That's okay. All right, so mitosis. What you're going to remember about mitosis is PMAT. Okay, so remember PMAT. Those are the stages of mitosis in the correct order. So and what we're going to do is write it, but we're going to draw a picture on the front, and then we're going to write on the inside flat what happens, okay? What they want you to be able to do is, if you see the events and written out, they want you to put them in the right order, okay? So you have to be able to click and drag in the right order. Or if you see pictures, you're going to have to be able to put them, arrange them in the right order, okay? All right, so this one's prophase. Pro is going to be our first, okay? So prophase, and we're going to draw that same cell that we ended up with. Well, going into G1, we're going to draw that same cell. So in G2. So in G2, what's the chromosome number we wrote out to the side? How much?
2. I'm at 4.
4N, right. So when you enter after G2, going into M, is a 4N cell. All right, what we're going to show though, in order for the chromosomes to separate, the nucleus actually has to disappear. Okay, so you're going to draw a nucleus that's dotted because the nucleus has to break down in order for the some equipment to be able to pull the chromosomes apart. So chromosomes, we don't see chromosomes until mitosis. Okay, so what we're going to do is open it up and let's put nucleus disappears or breaks down. We'll call it disappears, but you know that just means it breaks down so you can't see it, okay? And then after that, we're going to put chromosomes appear. Form/appear, because it was chromatin, and now it's wrapped around the histones and form these nice little X's. So chromosomes form or appear. And that's where we're going to draw them. Now, why do I want to draw them in 2 different colors? Where do your chromosomes come from? Where do you get your chromosomes from?
Your mom and your dad.
Yeah, your mom, your dad. So that's why we're using 2 different colors, is one represent dad's chromosomes and the other have to do mom's. Now, we would draw 46 of these things. We're not. We're only going to do 2 and 2, so we'll get 4. So let's do one big one and a little one. So I'm getting a little blue. I'm being really kind of stereotypical here. The blues could be mom's, the red one's dad's. It doesn't matter. But and then I'm doing a big red and a little red. So what do we call chromosomes that look alike and carry the same genes? Yeah. So you remember homologous chromosomes? So they're pairs. All right, so let's label, this is the nucleus, and we're not going to label any more of these. We'll just do the first one so that way you know. The X's we know are chromosomes. Now here's the equipment. Do you know what a lasso is? Like somebody makes a rope lasso? Okay, so there is these little stocky looking X things called centrioles, and their job is to shoot rope, which are these spindle fibers, over to lasso the chromosome and to be able to pull them. So they don't just move on their own. So what happens is these little centrioles, which look like little X's, their little protein structures, they're going to shoot some of these little spindle fibers out like this, and they're going to attach to the chromosomes. So they're actually going to do all the work of moving the chromosomes around. So the little X thing is centriole. And then the little line is a spindle fiber. And they're all proteins. So if we didn't have protein, there's another reason why protein's important. You would never be able to make any cells. You'd be one cell and never make any more. Okay. So that's what it looks like. So the inside, we need to put down something about centrioles and spindle fibers. Okay, so open it, and we're just going to put that they appear or they show up. Okay, all right, so let's open it up and we'll put centrioles and spindle fibers appear. Technically, the spindle fibers aren't there, but the centrioles already are. They just kind of move to the ends, but we're just going to say that they show up. Okay? All right, so M. M means middle metaphase. Metaphase, middle. Okay? So I'm going to make it a little bit smaller. All right, so metaphase, middle. The size and shape of the cell should be the same. The nucleus is gone. So there's no nucleus. We've gotten it out of the way. But the equipment's still in place. So these little centrioles are over here like this, but they have lassoed the chromosomes and pulled them to the center of the cell. So you're going to put your chromosomes, here's my blues, and they can be in whatever order, it doesn't matter. So if we had all 46, they'd all be lined up. And then here are the spindle fibers that connect to them. Like this. Okay? So metaphase, middle.
Yeah, maybe.
Okay, so let's open it up. So spindle fibers, I'm going to put S fibers, move, pull chromosomes to the center, say middle, or they may even say equator of cell. It could be any of those. Okay, because I'll turn that back. We could draw, you may see it drawn like this. Like an equator going wrapping around. So just because we made them go up doesn't mean that's how they always are. The cells are turned all which way. So that's kind of how they will leave it. They'll just line up there in the center. All right. And then let's stop there because of Edpuzzle. Okay. So anaphase will be next. Then we'll do we'll do the rest of it Monday. Okay. All right. So Edpuzzle. Put that away. Keep this in a safe spot so we can finish it up. So we'll put yours in. All right. And then we'll go over here to Edpuzzle. And does anybody need the code to rejoin at all? Everybody get in? Okay. That's not y'all. Craig, can you get in? Okay. We're just missing one. Hold on, there we go. All right. The cancer one is very quick, really quick. You can knock it out very fast. Yeah, the cell cycle mitosis one, there's lots of questions and it stops a lot. Remember at that puzzle, you can always rewind and go back, you can take notes. However, and we're gonna take the average of the two scores.